Welcome to the pharmaphorum podcast. This is web editor Nicole Raleigh, and today I'll be speaking with Brandon Pence, who was recently appointed President and COO of Fujifilm Biosciences, a company previously known as Fujifilm Irvine Scientific. Welcome, Brandon.
Thank you, Nicole. It's good to be with you today.
Now, the new company name encompasses Fujifilm Biosciences' evolution over the past 55 years. From its formation in 1970 to its recent expansion to serve broader life sciences communities and customers. It is also a nod to its scientific roots as a pioneer in the cell culture media industry with a core focus on innovation and quality. And today, Brandon and I will be discussing that field in greater detail. However, to kick things off, as listeners have come to expect, Brandon, perhaps you could share with us a little bit of an introduction to your own journey to today and what propelled you along the path to your current work with Fujifilm.
Excellent. Thanks, Nicole. I've been with Fujifilm now almost a year, but I've been in the life sciences space my entire career, which is coming up on almost 30 years. And it's remarkable for me to kind of look back over that time and not only see kind of the journey my career has gone through, starting out in R&D and moving into business management and business roles, but more importantly, the evolution and growth of the industry overall. I'm very fortunate to have started with an organization that was rooted in science. And perhaps it's appropriate at this point to mention the founder of the business I started with, Rex Spendlove. Rex recently passed away at the age of 99. He was a legend and one of the founding individuals for modern cell culture, creating a serum that was used in cell culture work, starting in virology, but that later became a critical ingredient used in the manufacturing of recombinant proteins and other biologics used in therapeutic applications. Rex founded the company High Clone Laboratories, which is where I began my career. And I was fortunate to work with some of the early biotech companies that were focused on biologics development. As I developed cell culture media and supplements from an R&D perspective, interacting with customers around the world, my love and interest for what they were doing propelled me into the business side of our company. I took over responsibility for product lines and ultimately the business side of our cell culture portfolio. And in that process, became intimately aware of and acquainted with what our customers were doing around the world, working closely with novel developers as well as CMOs in various locations. As I look back over my time and see what the industry has gone through from a supply side perspective, the High Clone Laboratories business became a part of Thermo Fisher Scientific in 2006. And from that time forward, it's been remarkable to see how quickly the growth of this industry has occurred. Over the last 28 something years, we've gone from biologics being kind of the novel new type of therapy to where they're now mainstream and often a first line of therapeutic choice for medicines to be used with patients around the world. It's interesting to see as we go forward with looking at some of the advanced therapeutic modalities, how the products we use have also evolved to meet the needs and the demands there. As my career journey progressed, I had the opportunity to leave Thermo Fisher Scientific and join what was then GE Healthcare, which is now known by the business name of Cytiva. And in my career, in that opportunity, I had the opportunity to work out of Sweden and was joined with colleagues there that helped kind of shape my professional experience in a way that probably wouldn't have occurred had I stayed in the U.S. throughout the entirety of my career. The time at GE Healthcare was remarkable. It was around 2015. And as I look back at that time, the industry was really in a fast growth trajectory. Our markets were growing in the low double digits. And as I think about that, it really set the stage for how our industry responded to the pandemic in 2020 and beyond. So I'm excited to see what the future looks like. I've had the benefit of working for large organizations like Thermo Fisher Scientific and GE Healthcare. And I find myself today with Fujifilm leading a business that is innovative and focused upon the next 30 years and where this industry is going to go. Fujifilm has seen the value of life sciences broadly. And if you look at the investments that we've made, I think it's $10 billion over the last 15 years in this space. Fujifilm is evolving as a company. and sees the value that they can bring to this industry. And I'm excited to be a part of that and lead the biosciences business.
Thank you, Brandon. So, if we set aside the sort of grander evolution at the moment and we return to, as I say, Fujifilm's foundation in cell culture dating back to 1970, just to expand a little bit more for listeners, Fujifilm Biosciences is a global full spectrum supplier to the life sciences market, and it provides products and services that assist customers in advancing healthcare initiatives with an expanding portfolio of applications supported that include life sciences and discovery research, cell and gene therapy, which we'll come back to, as well as the large-scale production of biotherapeutics and vaccines. The company offers a comprehensive, it's fair to say, portfolio of discovery and research reagents, recombinant growth factors and proteins, specialty chemicals, and critical assay materials, in addition to its robust cell culture media and supplement solutions in the US, as Brandon was alluding to. But let's focus today in this conversation, as I say, on Fujifilm's work in the cell culture media industry. Now, the cell culture media industry is currently experiencing significant growth, driven by the increasing demand for biopharmaceuticals, advancements in tissue engineering, and the rising number of research activities in cell biology. The market is projected to reach $11.7 billion by 2032, and this growth is fueled by the rising prevalence, unfortunately, of chronic diseases, but also by advancements in personalized medicine and the burgeoning field of regenerative medicine. It is an industry that is segmented by product type into several categories, and these include serum-free media, classical media, stem cell media, specialty media, and others, and each type of media serves a specific purpose and caters to different applications and research needs. So, Brandon, my question, I was wondering, even though you've given us a sort of preamble and mentioned the late Rex and his part in cell media, I was wondering if you could run us through a sort of wider, more generalized history of the field up to the point of where we are now, and also, of course, providing example of Fujifilm's expertise within that.
Yeah, I think, it's interesting, Nicole, as you look back and see how the cell culture space has evolved. At the very beginning of biologics development, it was still, kind of very foundational from a cell culture perspective of using classical media and serum to enable cells to grow and to produce the biologics of interest. As the industry grew and as we became much more aware of what the unique types of cells were going to be used, whether it's CHO cells, which are still the most prevalent cell line in biologics manufacturing today, or other cell lines that could be used from insect cells to HEK 293 cells, the unique requirements required unique product development. And in addition to that, regulatory agencies were guiding us to start to eliminate the use of animal products wherever we could. And while those legacy products of classical media and serum provided adequate performance, they also provided kind of the blueprint, if you will, of what nutrients were valuable and what nutrients may not be as necessary for certain cell types, which led to the development of unique and specialized formulations. So as you look today, you may think of classical media and serum as being capable of culturing multiple different cell types. Whereas when you move to a serum-free or even a chemically defined formulation, it becomes much more specialized. The formulations are focused upon a particular cell type. And even within that cell type, we see unique differences of the various clones that are used. Each cell, if you will, is as unique as the person that's culturing it. And because of that uniqueness, there are certain attributes that can be exploited for performance gains by nutritional requirements of a media formulation. And that's one of the areas that Fujifilm Biosciences is keenly focused on, is understanding, at kind of a molecular and genetic level, what that cell line needs, and then meeting the nutritional requirements of it through a unique formulation. The other thing I would add is that over that evolution of just not just the formulations changing with time, but I think that the performance has been influenced as well. We've been able to achieve far greater productivity titers than we ever have in the past. Today, it's not unheard of to have CHO cells that are producing at greater than 10 grams per liter. 25 years ago, if you were at a gram per liter, you were best in class. So we've seen not only the specialization of formulations and supplements that go into the cell culture process, but also the necessary output of increased performance as a function of becoming more specialized.
Thank you for explaining that for us, Brandon. I like this notion of each cell being as unique as the person who was culturing it. So that might be the takeaway for me from this conversation thus far today. So, when it comes to end to end life sciences capabilities to create an accelerated path towards greater patient access for advanced care, and here I'm sort of thinking of cell and gene therapy, Brandon, what can, in general terms, be done by industry? And let's go a bit further and touch upon AI, because given the world we're living in now, artificial intelligence has a role somewhere in all this, no doubt.
Yeah, I couldn't agree more. And I think that's actually one of the interesting crossroads that we're at right now. When you think about some of the advanced modalities like cell therapy or gene therapy, or even just the advances in CHO cell culture applications, we've got 25 plus years of data, of experience of growing these cells and interacting with these cells at both the research level and in production systems. I think the question for AI right now is how do we access that information rapidly in a way that guides us to the right solution? I think one of the opportunities from both a technology perspective intersecting with the advanced therapeutic modalities is that as we accelerate our interest in things like regenerative medicine and curing diseases in patients, historically that takes a significant amount of time to really understand, what the cell will do, how it will deliver the payload, how it will impact the patient, what is the right production system. I think AI has the opportunity to shorten that time frame, which allows us to get to patients faster. The other side of that is I hope that it will enable us to get to patients at a lower cost. And I think that's one of the advantages of AI right now. I don't know that we've got the solution today as an industry by any means, but I believe that there's enough investment in the space. And it's something that I was just recently at our scientific headquarters in Japan last week talking about. We look at that and see that the opportunity for AI to enable the science and the knowledge that we have today is remarkable. How we harness that is the focus of our R&D efforts. And I think that if we do this right, we'll see the development of things like cell and gene therapy not only accelerate, but come into manufacturing at a lower cost that will enable them to be utilized by patients around the world. So, yeah, I think your question with regards to how does AI do this is one that's being explored. I think there's no doubt that AI will influence it. I think it can influence everything from how we rapidly identify the right nutrient profile for the particular cell line, all the way through controlling quality and developing the right processing and manufacturing steps to deliver the medicine itself.
Thank you, Brandon. So you've been talking shortened timelines, lower costs, and you also mentioned the key word for my next question, which is manufacturing. So if we talk about manufacturing now, there has, of course, been a shift of late to the US for many large pharma during Trump's second administration, and Fujifilm Biosciences has facilities not just in the US, but also CGMP guideline adherent facilities in Japan. As you mentioned, you were at the scientific headquarters there, and also the Netherlands, as well as a media optimization center in China. So I was just wondering, has there been any effect at all for the company?
I think from a strategic perspective, we were well positioned for meeting this need, before it ever arose. And I think that that's one of the upsides to the way Fujifilm has looked at this. From the very beginning, they've diversified their landscape. And so as I come into the business a year ago and kind of take stock of where we were well positioned for really any disruption that could have occurred, whether it be geopolitical, social, things like the pandemic. We've got facilities on 3 different continents. We have the ability to supply from each of those. Many of our customers have qualified, at least 2, if not all 3 sites. And because our manufacturing processes are, harmonized between those 3 sites, we've simplified the way in which customers can do that qualification process. So while we've seen the shift in demand and the investment in manufacturing here in the U.S. as a result of the current administration's efforts, I don't know that it has necessarily changed what we've done because we were prepared for that to happen. I don't know that we necessarily anticipated, the current activities, but recognizing that those things could occur, we were prepared. And so I think from our perspective, we continue to invest in all three of our sites consistently. We haven't overweighted to one over the others at this point, and we feel very comfortable about the direction that our customers are taking, both in the U.S. as well as we still see a robust set of manufacturing requirements coming out of Europe, as well as in Asia, in particular the sites in Korea that are still very prominent in our space.
Thank you. So, in this state of being disruption ready, as it were, I have a final question for you today. You mentioned in your introductory section of today's conversation looking towards the next 30 years. So, as ever with this podcast, I like to finish things on a forward looking trajectory. So, Brandon, what do you think the future horizon looks like for those working in the cell culture media industry, given the ever changing times that we're in at the moment? And is it a dual aspect vision, perhaps?
Yeah, as I look forward to the next kind of 25, 30 years, I think it's going to be remarkable. First of all, I think the processes that we've developed that are utilized today will continue to be utilized into the future. And I think that we recognize that our industry is one that is intimately connected to people. What we do matters to the people that are receiving the medicines. And because of that, we are highly conscious of both quality and the ability to supply. And because of that, we're an industry that does not rapidly change, but does rapidly evolve. And what I mean by that is we aren't making changes and then hoping for the best or waiting to see what happens, but rather we're incrementally improving what we're doing today. Now, that has the application in current state of clinical developed molecules, as well as those that'll enter the pipeline 10 or 15 years from now. You've already seen over just the last 10 years an ability to not only scale up more efficiently in manufacturing, diversify regionally in manufacturing, but also respond rapidly because of the way we've set up our manufacturing processes, our supply chains. You think about the pandemic. We went to manufacturing vaccines within weeks, if not in months, having real vaccines ready for clinical evaluation. So we know that the industry can respond quickly because the foundation of the industry is sound. Now, as I look to the next 20 or 30 years, the things that could disrupt that, I think new modalities that are being developed, whether they're nucleic acids, whether they're the opportunity to use other viruses and vaccines in a different way to cure cancers. I think that there is the potential for cell and gene therapy to play a much larger role. We often talk about those as fast growing and large markets in the next 15 to 20 years. But we also know that the monoclonal antibody space will continue to be a prominent presence in the biologics portfolio as well. And so as I think about the professionals that will come up behind me in the next 20 to 30 years, I think what they'll find is that the expansion of biologics manufacturing and the ability to manufacture more efficiently in regions for patients in those regions will occur. I think we'll start to see the simplification of manufacturing in such a way that it'll be easier to establish multiple footprints versus one large hub that supplies the world. And that bringing manufacturing and bringing the medicines closer to the patients is something that I expect to see over the course of the next 15 to 30 years. And I think that will be a remarkable feat. It's one that will be challenging. It'll require novel innovators, contract manufacturers, bioprocessing suppliers, and regulatory agencies around the world to cooperate and collaborate. And I think in accomplishing that, we'll see healthcare become a leading force in driving positive outcomes in not just established countries, but in emerging countries throughout the world.
As you say, challenging, but that would be remarkable indeed. Thank you very much for your time today, Brandon. It's been a pleasure.
Thank you, Nicole. I appreciate the time.